I'm just, it's cold in the UK at the moment, so I'm going to ask you what the weather's like with you. I'm assuming it's nice.
Shall I start?
Yeah, I'm going to find out how I do the presentation. There's a button here now. So is everyone ready?
Introduce to you all. Here is a
great friend of mine,
Dr.
Matthew Bott
Matthew
from
Bott from
University of Berlin.
Berlin,
And
and
typically I'm talking
I'm
about
talking about
human
the
genome
human
mutation
genome
database.
mutation database.
So it's really very
It's really
difficult
very
to be
difficult to
so
be
formal
so
with that
formal
because
with that.
Matt is
Matt is
kind of
kind of
a person
a person
who,
who,
at least to
at
me,
least to me,
he knows
he knows
biology better
biology
than
better
I.
than I.
He knows,
He knows
definitely he is from
definitely he
computer
is from
background, but he
computer
knows
background, but he
biology
knows
better than me.
biology better than me.
And
And it is
absolutely a
absolutely a
pleasure
pleasure
working with Matt
working
because
with Matt because
we worked
we work
together
together
in many
in
projects.
many projects.
Currently, we are also
Currently, we are
working with
also
Matt
working
in
with
another
Matt in
project.
another project.
So I'm pretty
I'm
excited
pretty excited
to have
to
Matt
have
with
Matt
us.
with us.
So Matt, it's over to
Matt, it's
you.
over to you.
Oh, thank you, Anne, for that lovely welcome and introduction. It's really good to be here. I've worked with Bang for many years now, and we always have some good projects and good discussions, and it's always interesting working together. And I'm hoping that will continue. So thank you. So my name's Dr. Matthew Mort, and I work for the Human Gene Mutation Database, HGMD, which is a database of inherited mutations that underlie genetic disease. We release that as two versions of HGMD Pro and a public version of 3, but there's a bit of a delay on it. So I'm just going to talk about basically a lot of basic genetics relating to sort of disease and things to think about when we're interpreting sort of human genetic variants, so mutations and polymorphisms. Bioinformatics is a multidisciplinary area, so I think that would be where you need skill sets in sort of different areas, so data science, computer science, but also biology and genetics would also be really relevant. I think that's what makes it a challenging field to be involved in, is you require these different skill sets. A bit like Anne mentioned, it's important to have good biology and genetics, not just the computer science side of things. So I'm going to start with some introduction, but just this will definitely be a refresher for a lot of you, but that doesn't matter. I think it's just to get everyone on the same page. So in humans, what is a genetic variant? So you might hear sort of terms used like mutation or polymorphism or genetic variant, and they're types of genetic variant. In the UK, we're sort of moving towards not calling things mutation unless it's disease causing. So a variant would be classed as a neutral term. It doesn't mean that it's a disease causing variant or it's a neutral variant. There's just no connotation with it. It just means simply a difference or a permanent change between DNA sequences from an individual compared against the population. Inherited variants occur in your germ cells, so certain things are obviously inherited, and non-inherited variants can occur in somatic cells, so over the lifetime of an individual. Obviously, cancer springs to mind, which can be due to somatic mutations. So I'm not going to go into detail, but I'm just going to cover the basics. So we're talking
we're
about a
talking about a
variant, we're
variant,
looking
we're
at the
looking at the
population, and
population,
then we're
and then we're
taking an
taking an
individual, and
individual,
we're
and we're
comparing them
comparing them
against this
against this
population,
population,
sort of
sort of
the reference
reference
genome,
genome,
and we're seeing
and we're
what
seeing what
differences they
differences they
have.
have.
And
And
these
these
differences can be
differences can be
small or
small
they can
or
be
they can be
large.
large.
Um, so a
So a
small
small
variant would be
variant would
like
be like
a
the
substitution
substitution
choice where a
choice, where a
single
single
base has
base has
changed
changed
between the
between the
population and the
population and the
individual.
individual.
Population
Population
contributes to the
contributes to
wild
the
type
wild type
sequence. So
sequence. So
that's
that's
wild type
wild type
just
just
sort of
sort
the
of
most
the most
commonly
commonly
found
found
base at
base
this
at this
position.
position.
There's other
There's other
types of
types of
variants. You can
variants.
have
You
an
can have an
insertion where
insertion
you've
where
got
you've got
bases
bases
inserted,
inserted,
or you can have
or
a
you can have a
deletion where the
deletion where
bases
the
are
bases are
deleted,
deleted, and
that's an
that's an
encoding
encoding
region that
region
can
that can
alter the
alter the
reading
reading
frame.
frame.
So these
So these
variants,
variants,
there's lots
there's
of them
lots
in
of them in
everyone's
everyone's
genome,
genome,
and
and
it's
it's
when you're
when you're
looking at it from
looking
a
at it from a
clinical
clinical
perspective,
perspective,
we're
we're
trying to
trying to
identify
identify
which
which
variants,
variants,
it might be more than one, are responsible for the disease that the patient's got. And that's something in the UK, in our sort of like NHS, which is our health service that's free, we're increasingly trying to do. So we're doing a lot of screening on newborn screening to identify sort of a big panel of diseases early on, because if you treat them earlier on, you can sort of prevent and save money basically. So it's definitely an important area of bioinformatics. So other types of, so we've got variants. Now when a variant is in a coding region, so a gene, you can get different types of mutation because of the impact on the resulting amino acid sequence of the protein that arises from the gene. So a single base substitution or a point mutation could result in a silent change, which means there's no change to the amino acid that is at that position, a nonsense change, which results in the protein being truncated, and a missense change, where you just swap out one amino acid for an alternative amino acid. These are just the basics of different types of coding substitutions, and it's easy to see the impact at the protein level for these types. So they're often used quite extensively in NGS sequence analysis. So in this database, so we've got this HGMD, this mutations that are associated with disease. We sort of curate a lot of papers from the literature, probably about 40,000 published articles. Curators will have to read, interpret, and then record in the database. And what we notice is that the way a variant is described in the literature impacts, how we're able to incorporate that into our database. So it's really important that accurate variant descriptions are used in the literature. So if you're writing a paper, it's important you give an accurate description. And I'm going to give some tips of the best way to sort of present your variant data, which will be important if your work is relevant to clinical diagnostics and it enables sharing and comparison of your data with other data sets. You've probably heard of this HGPS, which is the Human Gene Variation Society, have produced recommendations, so nomenclature for standards of how to describe variants. And it may seem simple and straightforward, but when you get more sort of complicated variants, it's really important you follow the recommendations, because it makes it easier for others to use your data, and it makes it easier for your data to have clinical utility in other aspects. So we're just going to start with, you've got a variant, you've done some NGS and next generation sequencing, And that variant can be described in different ways. And commonly, when you do NGS sequencing, you'll get your data at the genomic level. And that's where you can describe your data at the genomic level, and that's a really good way to describe it. But also, because we've got genes in the genome, you can describe it at the gene level. So that would be used in sort of reference sequences, these that start with this NM prefix. And you can also describe it at the protein level. And when we curate HGMD, we notice that of mutations or variants that are reported in the literature, around 15% are either inadequately described, so there's not enough detail that allows us to locate the mutation, but also they have errors. It's easy to have a typo in a paper. It happens quite commonly, 15%. So what we would say is, if you can describe the same variant at two different levels at a minimum, even if it's just gene level and protein level, that is really useful because it allows you to double check the protein level description against the gene level description. So in terms of best practice, I would say use at least two different levels to describe the variant. If you're publishing your work, that will allow for people to check the data, and if there's any typos, they can be picked up. If you're using reference sequences or reference protein sequences, include the version number, so the .6 or the .2, .1, and that's useful because these sequences can change between versions, and it just clarifies where your variant is. So I'm not going to spend too long on this, but a reference sequence, you might be saying, what's that? And it's basically a digital file, so a text file with an idealized version of a DNA sequence in an individual, or it can be a different species as well. A variant then can be described against that reference sequence. So this reference sequence is a bit like, you can think of it as a map, and you're saying, my variant I found is different from the map at this position and that position. And that's where you see these different prefixes being used in relation to a reference sequence. So you might get a C dot, a G dot, an N dot, and a P dot, and they just refer to what level the difference is occurring at. So a P dot would refer to the protein sequence, and you're starting at the first amino acid, the methionine being a 1, and that's your reference point. There are other reference sequences you can use, like Ensemble, which is a good data set to use for reference sequences. We prefer to use NCBI's NMs. We find it a bit more conservative and they're more stable, so we prefer them, but there's nothing wrong with using Ensemble either. This is just an example of how you find a reference sequence. You've probably seen this before and downloaded these files and used them in your own analysis, but the version is important. So if you're publishing a manuscript, try to include the version number, the point something. That's an important bit of data to include. Now we've just got some examples of how variants can be described. So we talked about G dot, which would mean the genomic level. We talked about C dot, which would be the coding mRNA. But also you may have variants in cis or trans. So cis is just where you've got two variants on the same chromosome, so they would be within the same sequence, you've got two changes. And I think this is something we don't handle very well in bioinformatics. We sort of treat the mutations individually when we should be looking at multiple changes and the impact of the multiple changes at the protein level, the impact that has. and this doesn't always happen. With some of the newer NGS platforms, you can identify whether a mutation is in cis or it's on the opposite chromosome. And this information is useful, especially in diagnostics. It's something to be aware of when you're annotating mutations. So we talked about these HGVS descriptors, so how to describe your mutations. And there's some bioinformatic tools basically online, and this is the Mutilizer 3, which is a free tool, and it will help you with your descriptions. So you can put in a HGVS description and sort of validate it, and it'll say, yes, that looks right, you know, a G is present at this position. It will also convert, so you can convert genomic positions into HGVS and vice versa, and it's a really useful tool. And I think if you're publishing any mutation data, there's a bulk validator where you can validate a sort of set of mutations or variants, and it's useful to use a tool like this just to check if everything's correct. We curators at HGMB use it to sort of find any issues with mutations that have been reported. And I think if you can identify them before you publish, it's a good time point to do so. So I'm sure a lot of people will have heard of VCF format, and this is when you're doing your next generation sequencing, this is pretty much what you'll get from the machine. You'll get a text file, it's tab delimited. It's a really good format to use computationally because there's no sort of wishy-washy, you know where you are with it. You've got the reference base and then you've got the alternative base, the variant. It is hard to describe sort of large deletions and large sort of genomic rearrangements in this format, but for smaller variants such as substitutions, insertions, and deletions, it's really useful to use, and it will be a file that you will come across. So you will be aware of it probably already. There's a lot of tools out there that can take a VCF file and do different things with it, merge them, filter them. VCF tools is a great resource for doing that, and if you haven't heard of that one, I would check it out, it's really good. Right, so we sort of covered the basic groundwork of genetics and variants. So what I think we need to get across, or what I'm seeing in the literature is when we're prioritizing variants, so we've got a patient, we don't know the mutation that's disease causing, we do an NGS sequencing, and we're trying to find the disease causing mutation. So often in that setting, in the UK, a clinician or a doctor would say to us, you know, we want to find the disease-causing mutation, and I want a small list of like 1 or 2 mutations that are disease-causing. And I have to tell the doctor and say, it's not always possible to do that. I can prioritize, but I can't. It's not a definite. So when we're doing this prioritization using NGS, it's all about probabilities. So it's like, this is the likely mutation, but without doing further functional work in a lab, I can't say for certain. So that's a concept to think about, probabilities when we're dealing with filtering and prioritizing variants. So I think one idea or concept is that when you're looking at someone's genome, all variants have the potential, at least, to either on their own or in combination with other variants to be disease-causing. So we have to keep that idea in mind when we're doing this filtering. So when you're developing an NGS pipeline to filter and prioritize the large number of variants you get from a genome or an exome screen, you try and filter out things, and you can use mutation types as a rough filter. And by mutation types, I mean if a mutation is truncating, so it's a nonsense mutation, that would be a high probability to be disease-causing. But obviously it depends on the gene it's truncating as well. So it's about the mutation type and also the gene that the mutation occurs in. And we can think of these different types having different probabilities. So on this slide, I put probabilities, but these are just rough. It's not, they're just arbitrary probabilities. So we can think that a truncating mutation is more likely than a missense mutation to be disease-causing in general. There's always exceptions to this. And a missense mutation, one that changes an amino acid, is more likely to be disease-causing than a silent mutation. And so on and so on. So a mutation outside of the coding region is less likely to be disease-causing than a mutation within the coding region. But it's not impossible, so that's what we have to keep in mind, that it's not impossible that a non-coding mutation is causing disease. It's just less likely. And I think with a lot of the pipelines that I've sort of seen in action, they might completely filter out sort of non-coding mutations or other classes of mutation, like same sense, which you have to prioritize, but if you don't find good candidates, then it still could be within non-coding or even a same sense mutation, and that's one aspect I'm trying to get across. There's nothing wrong with using variant types to prioritize, but still keep in your mind that it's possible that a non-coding or even a same sense mutation could be disease-causing. And also, the other thing, before I move on, is that it could be more than one variant is acting together to be disease-causing. So when we're profiling these mutations, we're just looking at a mutation on its own, in isolation, and its impact. But especially if you get multiple changes within the same gene, it could be the combination, or even multiple mutations within different genes, and their impact that may be disease-causing. So having one mutation, you're fine. If you've got both, you may be entering a disease state. That's always a possibility, but it's something we don't cover very well, I think, in the current bioinformatic methods. So I'm sort of highlighting this idea about probabilities. And we're saying that a nonsense mutation, one that truncates a protein, is more likely to be disease-causing. But it doesn't mean other types can't be. So a silent mutation, that is one that it doesn't change the amino acid. Now straight away, someone might say, well, that can't be disease-causing. It doesn't actually change the amino acid. That's not strictly true. They might be silent with respect to changing the primary protein sequence, but they're not always neutral. And there's been a lot of research on this, so we see that these silent variants commonly end up negative selection. So the selection force is acting on them. So that hints that they may be functional, or sometimes functional, not always, sometimes. And we know that usage of a different codon in the sequence can alter the accuracy of mRNA translation. So it's obvious that genes prefer certain codons. And if you use the alternative codon, which still encodes the same amino acid, you might get more errors. So it might reduce your levels, your gene expression levels. We also know that different codons can alter how an mRNA folds, which may impact sort of how much levels of protein are translated. A silent mutation may also disrupt splicing, because it could be close to the splice site, it could be the last base of an exon, but it could also disrupt other splicing elements, such as splice enhancers, and lead to missplicing. Another thing we know that a silent mutation can do is it can affect the speed of translation. So some codons are translated into the protein sequence faster. And this, when you've got a large protein, like collagen, it will fold slightly differently if translation occurs quicker or slower. So that can affect the final shape of a complex protein. For a small protein, it won't make much difference. A silent mutation could also disrupt microRNA binding sites or other sort of transcription factor binding sites that are in the coding region. So you can use mutation type to filter, and that's a good idea, but just be aware other mutation types still could be disease causing. So if you can't find your disease causing mutation, you need to look further, and it might mean looking at silent mutations or non-coding mutations. So I think if you're doing an NGS screen, it's a good idea to focus on the coding region, but don't ignore non-coding. There was some research looking at developmental disorders, and they looked at the coding region to identify developmental disorders in children, and for about 60% of cases, they found mutations in the coding region, sort of suggesting that 40% may be outside of the coding region. So it shows you that the coding region is important, and probably the majority of disease mutations will be in the coding region, but they could be outside of that area as well. When you're looking at the coding region, it's easy to see the impact and sort of put towards evidence for a mutation to be causative. So if you look at a truncating variant, you've got a good case. I think this mutation is disease-causing because it truncates the gene responsible for the phenotype, and that makes sense, and there's nothing wrong with that. With non-coding, it's hard to interpret that data because you can't see the impact directly, as it were, based on the biology. So you find a non-coding variant and you say, well, I don't know what this does, but it could be interesting, but I don't know. It's in a conserved region, but I don't know the actual functional impact of it. So you might be asking, how does a non-coding variant impact and cause disease? How does a non-coding variant have a functional impact? And often, so a non-coding variant is due to a change in gene expression, typically. Not always, but typically. So if you get a non-coding variant in the promoter of a gene, it could reduce gene expression and cause loss of function, but could also increase gene expression, and you would class that as a gain of function mutation because you're overexpressing the gene in question. A non-coding variant can also disrupt splicing and result in sort of expression of a new sort of transcript of protein that's not typically present, and that would be a gain of function mutation. How would it do that? So it could do it via transcription factor binding sites, disrupting those. There's also mechanisms of where the chromatin folds, sort of non-coding variants can disrupt that, especially larger ones. And these can affect the domains involved in the folding of the chromatin, and it can affect gene expression again. Non-coding variants in introns can disrupt splicing, especially close to the exon-intron boundary. And in general, our sort of the regulatory code of the genome, the sort of non-coding bits, is still poorly understood. So we don't understand everything or even much about regulatory elements and how they affect gene expression. And especially they think there's a sort of a long range impact as well. So you could have a variant at one place in the genome and it's affecting not the nearby gene, but a gene that's miles away, very far away from it. And it can still regulate that gene because in the 3D sense it's close by. And this is something we don't fully understand. So when we sort of prioritize the mutations, we need to take that into account. We don't understand much about the regulatory code of the genome. There has been projects to try and sort of decipher what's going on in the regulatory code of the genome. You will have heard of the ENCODE project, which was quite an interesting project. So most of the genome is non-coding, 2% is coding. And of this non-coding ENCODE project, roughly around 80% of the non-coding bits, we could link it to some biochemical function, which is interesting and makes sense because when I first started doing genetics in 2000, it was more the other way around, so we would use the term junk DNA for the non-coding regions. And what we're seeing now that is there's probably not that much junk DNA or DNA that has no purpose in the genome. If it's there, it's probably got some sort of use or function. There's so many different types of regulatory elements that are important. It'd be really interesting as bioinformatic tools develop and be able to sort of assess the impact of variants in non-coding regions. It's just something to be aware of. So for this then, I was trying to assess how much of genetic disease is due to non-coding variants. What was interesting. Obviously, these are just rough numbers, but as that study I mentioned earlier, where 60% of patients, 60%, they could find a coding region variant that sort of was causative of the disease, leading to them to not find variants of 40% of the family studied. So that suggests that maybe, roughly, maybe the upper bound is 40%. Of disease-causing mutations may be due to non-coding variants. In our database, we've got about 10% of the total variants are non-coding. The majority of those are sort of intronic variants, which would be disrupting splicing. And there's a definite bias in the literature, so people will look in the coding region more, so they're more likely to find coding region mutations. So a rough estimate, I would say, is 10 to 40% of human genetic disease is caused by non-coding variants. This will differ based on the disease, and I think we'll find that some diseases are sort of biased towards non-coding, where other diseases are biased towards being in the coding region. And that's just a key point I think to take away. We should focus on coding region variants, but don't completely exclude non-coding. And if you're developing a pipeline, consider sort of a two-tier system where you sort of focus on the coding region, but then you also profile and annotate the non-coding variants as well. Don't completely exclude them from your analysis. I've talked a little bit about splicing of genes. Probably should have mentioned the genetics underlying that earlier, but it's basically on your reference genome, you've got exons and you've got introns. And the introns are basically cut out or spliced out before your protein is made, and then your protein consists of all the exons that stitch together and then translated. Probably familiar with this process anyway. Obviously, if you get variants at splice sites, which is the intron-exon junction or close to there, they could disrupt this process, and that would lead to missplicing and the generation of transcripts that you wouldn't normally see, novel transcripts. Now, what the interesting part of splicing is the same gene can result in multiple different transcripts, which can then be translated to different proteins. So, if you're not aware of alternative splicing, it's a really important concept. It's how you get diversity at the protein level based on one gene. So one gene could code for 11 different proteins, which could, some of them will have related functions, but the proteins that arise from alternative splicing could have different functions as well. So the impact for your variant then is also important, because if we look at these three proteins, A, B, and C, so one of the main differences is exon 3 is missing from protein B. So it doesn't have exon 3 in there. And it's still a functional protein, just might do a different job. So if we're profiling a mutation in exon 3, it's important we don't use transcript, the protein B transcript, to sort of evaluate it, because we would say it was non-codable. Whereas in protein A or protein C, the variant from exon 3 could be like a truncating mutation, whatever. So it's important when we're doing annotation, maybe bioinformatically, that we're using the right transcript, or we use all the transcripts. With alternative splicing, what you'll find is some transcripts may not be expressed at very high levels. So protein A may be only expressed during, embryo, but as an adult, it's just B and C. So this sort of information is useful. So if you're looking at a brain disorder and you see that protein C is expressed highly in brain, but protein B is not, you would prefer, or you would prioritize, annotating a mutation in terms of the functional impact in protein C. And that's an important sort of fact to sort of add in. If you know the disease you're looking for and the tissue, you can use that information to prioritize your mutations better, because you sort of prioritize the ones that are highly expressed in the tissue of interest. And that's an interesting concept, and it's something that's really important, because you may miss the disease-causing mutation if you exclude a transcript, and you may miss it if you don't use the right transcript based on expression levels in your tissue of interest. So to get across how important alternative splicing is, it happens a lot. Pretty much 95% of genes with exons and introns undergo alternative splicing, so almost all genes. On average, you get 7 different transcripts and potentially 7 different proteins per gene on average. Some will have more, some will have less. I've mentioned about differential expression across tissues and developmental time spans. So depending on the age, it may be that this is a transcript that's expressed only as an adult, or it may be a transcript that's only expressed developmentally early on. When you do your variant interpretation, you might need to consider transcript selection and tissue expression. That's one way you can sort of tweak your pipeline to include that information. So here's an example of how alternative splicing, using that knowledge and that information, improved clinical diagnosis. So we had one study, they looked at about 300 genes on 3 neonatal epilepsy panels. But each panel, so these NGS panels, they only considered one transcript for the gene. So even though the gene had multiple transcripts, they only considered one. They picked one transcript, possibly just the longest transcript. And this was a commercial panel. They found that of the 300 genes, 30% of the genes they were alternatively spliced, and they had other transcripts with alternative coding regions that were expressed in brain tissue. We're looking at sort of brain disorder, epilepsy, and they found that when they used the extra transcripts, they found that they picked up 4 additional variants that were pathogenic that they didn't find before. So for this panels, it was important to, if you include more transcripts, you might improve your clinical results basically. So this issue of alternative splicing can improve your results. So it's just something, again, to be aware of. This is sort of an idea that I have to get across to clinicians. So when we're looking at an NGS panel, we're trying to find, we're looking at an NGS screen, we're trying to find a needle in a haystack. A clinician might just want one mutation, but the results in practice are probably going to be prioritized lists of groups of mutations, where we can't say for definite which is the causative mutation, but we sort of think, like, it's either in this set or it's in this set. And that's to do with these probabilities when we're doing this type of work. So finding the disease-causing variant is important for clinical treatments. Patient outcomes can depend on them, and by getting it wrong can lead to bad outcomes for patients. So it's important we do a good job and we are thorough when we're doing this type of work. The ACMG Society present guidelines for variant classifications, and they can sort of classify them into 5 different tiers: pathogenic, likely pathogenic, uncertain significance or bus, likely benign, and benign. And if you are working in this area, you should read up on these classification guidelines. And they use sort of 28 criteria to do this. So they look at population data, computational data, so that's sort of bioinformatic tools to say if mutation is disease-causing or not. Functional data, so that would be stuff in a lab. Segregation data, which is looking at families and does the mutation occur always in someone with the disorder. De novo data, looking at mutations that have newly arisen and not in the parents. Allelic data, other databases, and other things as well. So they apply weighting. So it can get quite complicated, but basically they look at these different lines of data, they apply a weighting to make, and this is all to do with classifying mutations in these classes as probably disease-causing, likely, uncertain, benign, or likely benign. So this chart is what clinicians in a diagnostic lab would be looking at to make a decision. So they'd look at population data and they would say, if this variant they found is in a population at a high frequency, it's unlikely for it to be disease causing. So that's strong evidence that the mutation or the variant is benign. Benign just means not harmful, so not pathogenic. I'm going to go through these quickly on some more slides now. So this ACMG classification, it's all about coming up with this label, disease causing, uncertain, significant, benign. There are tools that will do part of this classification, but they won't do all of it. So Intervar is a good tool for doing this, but it will only consider the sort of bioinformatic aspect. So it will look at population data, it will look at bioinformatic tools that make predictions, but if you have additional data like segregation data, de novo, it won't include that. So you need to add that in manually or in your own scripts when classifying mutations. So we can just look at the types of evidence for pathogenicity. It's along the lines of these weightings that we've given. Population data, so that's something to be aware of. If you've got a rare disease and the variant you're looking at is common, it's unlikely that that common variant is going to be disease causing. That's the general idea. If you've got a common disease, say diabetes, that may be at a population frequency of 1%, then you're more tolerant to having a higher frequency variant and it's still being disease causing. So when you're using population data to sort of as one line of evidence, consider how common the disease is and how common your variant is. Typically, you'd be looking at the 1% or 5% levels. So my mutation's found at 5% in the population. It's a rare disorder. I don't think it's pathogenic. And so population data is useful information to assess pathogenicity. Nomad is a really good database to get that data from. It started out as the exact data, and it was just sort of exomes, and then they added in whole genome data, and it became Nomad. You've got data from 200,000 individuals. You can look at different populations as well. I think if you know the ethnicity of your patient, you should try and tailor that population you look at to the same ethnicity. I think that's an important thing to do, and it will improve your results. Nomad's the main database we use for population data. So another line of evidence is these computational and predictive data in silico, basically tools. They're really useful because you can profile a lot of mutations using them. Some of them might give you the possible impact of the mutation, what it's going to do to the protein. Very important tools in bioinformatics to classify mutations. There's a big list of them here. You've probably heard of quite a few of them, and there'll be some additional ones you might use yourself. At HMD we use CAD a lot. We like that because it takes BCF format and it can profile all types of mutations, coding and non-coding. But there's many other tools as well, especially for non-coding. You could look at sequence conservation. That's a good way to sort of analyze non-coding variants. There's a lot of tools that are just for missense mutations. They're useful to use. Often these in silico tools will make predictions using the same sets of information. Not always, but generally. So they will look at evolutionary conservation of amino acid or a nucleotide location. They'll sort of incorporate the context within the protein sequence, and maybe the biochemical consequence of the amino acid substitution. In general, it's not all, but most tools will make similar predictions. So if you're using 10 different tools, you will get better results using a consensus of those predictions, but it won't improve much. It might improve by sort of 10 to 20%, your prediction accuracy. It's generally because these tools are similar underneath. So because they're similar with some differences, they make similar predictions. If you think about them roughly as being like 65 to 80% accurate when making predictions about mutation being disease causing, what we do know is they tend to have low specificity, so they over-predict variants as being harmful. So while they are a good line of evidence, if you're doing something in a clinical setting, you should never base it solely on an in silico prediction. And what we're seeing in HMD is that a missense variant, so amino acid substitution, could have a mild effect, but still be disease causing, and they're not really reliable at identifying small effects. So for some diseases, you get big impact on protein and it will pick those up. But if it's just a subtle effect on the protein, but still disease causing, these tools are not really effective at that. So that's why we must rely on multiple lines of evidence. I say functional data, if you can do it, is really good data. This is basically stuff in a lab. You can understand the functional consequence, and it might provide useful information to help with treatment or diagnosis. Sometimes it can be difficult to interpret functional data, so how far from the sort of normal wild type function do you have to go before you class it as abnormal? But still, once if you've done your prioritization, if you can do it, I would say doing functional work is really a good idea, but it's not always possible due to time and financial constraints. There's lots of different functional data you can do. Look at the different levels. With the DNA level, you're introducing a mutation with CRISPR. You could be looking at the protein level. I think it does add weight to your results, but it is expensive. That's the only thing, that's the big drawback, maybe the time consuming. So I'm not really going to touch much on what other functional work you can do, but consider genome, proteome, transcriptome, epigenome, and even the microbiome. We're seeing that the microbiome interacts with the immune system quite a lot, so you could have a genetic variant, and when your microbiome sort of goes a certain way, then these interact to cause disease. So your genome can interact with your microbiome and cause disease as well, especially immune system disorders. And that's one interesting area that there's a lot of work being currently carried out on. I talked about segregation information. So if you've got a family and you've got pedigree information, so you know like the disease status of parents, grandparents, brothers and sisters, and you can sort of do this segregation diagram and information, that can provide evidence because if a variant segregates with the disorder, so every person that's in the family that's got the disorder has the same variant, it could be linked with it means that it's possible that variant is disease causing, but it doesn't exclude the fact that it may be linked to another variant because variants can travel together. So one variant can travel with another one through a family, but it does help. That information does help when you're trying to narrow down. So if you've got family information, that's really valuable. Basically what I was saying again, if a variant doesn't segregate, that means there's strong evidence for the mutation being, or the variant being benign or neutral. So if the variant's in one person in the family, and they've got the disorder, but it's not in the other person in the family who's got the disorder, that's stronger evidence. You would say it's probably not the disease-causing variant, because they both would have the same variant. So it's just something to consider again. De novo variants, so you've probably heard of de novo variants, so it's basically a new variant that's not found in the parents. And when this is useful is when the parents, or no one in the family above the offspring, are affected with the disorder. So the parents haven't got the disorder, and you get an offspring with the disorder, then potentially it could be due to a de novo variant, a new variant that's not in the parents. And on average, in an exome, every person would have 3 de novo variants typically. So if you know the parents haven't got the disorder, and it's not a recessive disorder, it's a dominant disorder, then de novo, you would look at the de novo mutations first. So that would provide evidence for that. Especially if the de novo variants were in a related gene or a gene of interest. We see this with autism. There's been a lot of studies with de novo variants in autism. So some disorders are sort of typically characterized by de novo mutations. And if you know that information, you can make prioritizing easier. And so for the last part, we're going to look at databases as useful resources to help and aid in the assessment of pathogenicity. There's a lot of information in databases that you can leverage in your pipelines. So we talked about population databases, so you can get the frequency of your variant. Maybe your variant's not in these population databases, and that's a rare variant's more likely to be disease-causing than a common one. You can look in disease databases like ours, HGMD, to see if that disease-causing mutation has been reported before. That's a good line of evidence. There's also some functional databases where they've got sort of functional data, and you can query those as an additional line of evidence. I think one of the things when dealing with databases and other resources, evaluating the data quality for users is difficult. Like how often is it updated? You could look at, is it sort of manually curated, user submitted, or is it sort of automatically generated, maybe using AI? What degree of the database data validated? What the source of the data? So these are just things to think about when you're assessing a resource. Not all resources will be the same quality, but it's tricky to evaluate the quality until you start using the resource. So with HGMD and other disease databases, they can be used to assess pathogenicity. So if you've got a mutation, has it been reported before? You check the database, it's already reported. So that gives you sort of a good line of evidence for this variant you found being disease-causing. It can also give you an idea for the disease in question, the types of mutation that you get for a particular gene, because you see that some disorders are often caused by nonsense mutations over missense, and vice versa. So if you can look at the mutational spectrum for a disorder, it might help with your prioritization, because you could say mostly the disease-causing mutations are in exons 3 to 5. So we're going to focus on that region in our prioritization. So knowing the landscape regarding a gene and the mutational spectrum is important, and it can help you do that. So that's another way. The more you know about that gene and how it causes disease, it can give you insight into your new patient with the same disorder, and it can help you prioritize better. There is other databases. There's ClinVar if you're interested in disease-causing mutations as well. That's freely available, so you can just download that. User submissions are included in ClinVar. There's also data that's not published. I would say it depends what you want to do. ClinVar can be a good data source. Just be aware of its limitations. I'm mainly going to focus on HMD because that's our database and that's sort of what we're familiar with. The number of mutations that have been reported in the literature has literally tripled since 2010. As with anything, because of sequencing costs come down, there's a lot more sequencing going on, and we're seeing that in terms of curation workload. We only include germline mutations, so we don't include somatic mutations. We only look at the nuclear genes, so we don't include mitochondria. We've got over, currently now we've got 400,000 mutations from about 15,000 genes. And we look at there's over 100,000 articles that someone has to curate. And we look across the 3,000 different journals. So it's a lot of journals and it's a lot of work. We do have a free version you can access, which is basic, but it's just 3 years out of date. But it still can be useful for your work. In HMD, we divide mutations. So we talked about the ACMG guidelines, they have probable pathogenic. We come up with our sort of categories before ACMG. So we go with a DM, which is a disease-causing mutation, and a DM question mark, which is likely disease-causing, but with questionable pathogenicity. So there's some doubt. This would be where the authors say, we think this is the disease-causing mutation, we're not sure. We do have other types which are at higher frequency, so a polymorphism would be found at 1% frequency or above, and these are disease-associated polymorphisms, and disease-associated polymorphisms which support functional evidence, which would be a DMP. And then an FP would be where they've done a functional study on a polymorphism, but there was no disease linked to it. So it impacts function, but currently there's no reported disease. When we're comparing different databases, we can see between ClinVar and HGMD, there's a big overlap, but ClinVar captures 24% of HGMD mutations, with about 76% of HGMD specific to HGMD. It's 40% of ClinVar that's not in HGMD, and these represent mutations that have been submitted by clinical labs but not published in the literature. And because we only include published mutations, we don't include them. We're sort of coming to the end now, but I think the key points that I'd like you to think about are that identifying a single variant responsible for disease in an exome or a genome screen is tricky. It's about prioritizing and probabilities. So think in terms of probabilities when you're dealing with this type of data. The second point would be, based on probabilities, it's likely your variant will be in the coding region, but don't completely exclude the non-coding regions of the genome, because you can have functional variants there. And I think the third point would be, in silico tools are really useful, but they're only predictions. So results with a clinical consequence should never be based solely on an in silico tool. Use multiple lines of evidence. And saying that, if your in silico tool says this mutation is harmless or benign, but your other evidence, your sort of real world data, your segregation, maybe your functional study says it's harmful and disease causing, then there's nothing wrong with overriding the in silico tool prediction, because it's just a prediction. So I think those are the three things to think about overall when doing this type of work. And I've just got to acknowledge everyone at HGMD and QIAGEN for supporting us with funding, and thank you to Anne for all his help and all his for inviting me here today. And it's been really good to talk to you all. And that's it.
Thanks a lot, Matt. It's a fantastic presentation and a big round of applause.
Thank you.
Matt, I think there are a couple of questions for you. The chat box. Yeah. So the first one is there any relation between the position of the codon chain with the pathogenicity?
I'd say yes. So obviously with the codon that codes for amino acid, the third position is the wobble position, so it's less likely to code a different amino acid. So typically be the first and second positions. But when you're considering splicing, it can be any three of the positions. So that would be, if you're looking at the protein level, yes. If you're looking outside the protein level, and looking at impact splicing, no, it could be any position.
There are a couple of other questions which are how can I detect the same sex mutation at the protein level and distinguish them?
Can you explain that in a bit more detail? Or say it again now.
Okay. Then there is another thing that is written like how can you differentiate between no mutation and silent mutation?
So when you've got your VCF, you can run it through tools to annotate, and they'll sort of annotate against reference sequence, and it will produce all like ANOVAR, it's useful tools. So you take a VCF file, and it will produce the CDOC description all transcripts is one of the options it has. Then you can filter out and see if it's a missense or a same-sense mutation from your VCF.
And does alternative splicing increase the chance of mutation?
Alternative splicing impacts. So it impacts your interpretation of mutations, because you've got one gene that can code for, say, 10 different proteins. So when you're annotating that mutation, you have to consider the different proteins. So normally we would just conform protein or mutation. But one mutation can arise in multiple proteins in different contexts. And that's the difficulty. So in that study, when they looked at, they took just one transcript from one protein and mixed the others, they didn't find all the disease-causing mutations. So it's saying broaden your scope to improve your results.
And there are other two questions. How can you change the therapeutic aspects with respect to disease variants? I think I answered this question.
Um,
And
I think it's
and
asking if you
if you
know
know
the
the
disease
disease
causing
causing the
mutation, you
mutation,
sort of
you
know
sort
in
of know
which
in
gene
which
it's
gene
in,
it's in,
and you know in
and
the
you know in the
function
function
then if
then
that
if that
information is
information is
available.
available.
So we
So
know
we
that
know that
different
different
therapeutic
therapeutic
options
options
are
are
available
available
depending on the
depending on the
gene
gene
that's being
that's been
impacted.
impacted.
And this is
And
more
this
of
is
a
more of a
complex
complex
disease.
disease.
So if you've
So
got
if
an
you've
immune
got an immune
system
system
disorder,
disorder,
there's lots
there's
of
lots of
different
different
genes
genes
involved in the
involved
immune
in the immune
system.
system.
So if you're
So if
knocking
you're
out
knocking
gene
out gene
X, it may be a
it may be a
different
different
therapy than if
therapy
you're
than if
knocking
you're
out
knocking
gene
out gene
Y,
Y,
because they have a
because they have
different
a different
function.
function.
So
So
that's what
that's what
questions
questions
asking. I
asking.
think
I think
it's
it's
important to
important to
know
know
the
the
underlying cause,
underlying
not
cause,
just
not
the
just the
patient's got
patient
a
has got a
disorder. If you
disorder.
know
If
the
you know the
underlying
underlying
cause,
cause,
then it
then it
helps
helps
target
target
which
which
drug
drug
choices or
choices or
treatment
treatment
options to
options to
apply.
apply.
is there any possible tool available to detect the effect of snakes?
Considering the whole
Considering the whole
gene.
gene.
You know.
Yeah.
Yeah.
So that would
So
be
that would be
paired with a
paired with
really
a
good
really good
one.
one.
You can use
You
your
can use your
VCF
VCF
files, your
files, your
input,
input,
and it will
and
look
it
at
will look at
all
all
elements of
of
change,
change,
and it will
and it
cover the
will
whole
cover the whole
genome, but it will
genome, but it will
prioritize
prioritize
them
them
within the
within
same
the same
list.
list.
So
So
it can be
it can
really
be really
useful if you've
useful
got
if you've got
non-coding
non-coding
stuff,
stuff,
same
same
sense
sense
mutations,
mutations,
silent
silent
mutations,
mutations,
and
and
missense and
missense and
nonsense.
nonsense.
Really
It's a
good
really good
tool, I
tool,
think.
I think.
CAD,
CAD,
that is.
that is.
It's on the
It's on the
slides.
slides.
Okay, yeah. And there is another interesting question. Though I believe it is not directly related to bioinformatics, how to amplify two unique genetic transcripts of a single gene using one pair of primers?
That
That
sounds like
sounds like
a
a
lab-based
lab-based
question. I'm
question.
not
I'm
sure
not
I can
sure I
answer
can
that.
answer.
That's not related to bioinformatics, I believe.
I think
I
there's
think there is
ways
ways
from
from
my
my
experience, there's
experience, there's
ways to
ways to
sort
sort
of
of
increase the
increase the
transcription and
transcription and
translation of
translation of
specific
specific
transcripts.
transcripts.
And primer choice
Primer
would be
choice would be
important in
important in
that.
that.
But
But
you'd have to
you'd
look
have
at
to
the
look at the
knowledge
knowledge
of
of
the,
the
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 alternate
alternate
splicing and
splicing and
transcripts that are
transcripts that are
present.
present, and
You'd probably
you'd
do it
probably
on a
do it on
case
a
by
case
case
by case
basis, I
basis, I
suppose.
suppose.
Yeah. And there are some questions because most of the participants are students. So they're like, is there any good tutorial on this skills for bioinformatics? Can you suggest any website for this beginners for the bioinformatics?
I
I
haven't,
haven't.
there is a
There
lot
is
of
a
good
lot of good
stuff
stuff
out
out
there for
there for
bioinformatics.
bioinformatics. I'm
I'm trying to
trying
think
to
of one
think
that
of
I've
one that
seen
I've
was
seen
really
was
good.
really good.
It was an
It was an
American
American
university, but I
university,
can't
but I
remember
can't
which
remember which
one.
one.
Um,
Um,
I think
Coursera.
Coursera.
Coursera has
Coursera has
some
some
good
good
stuff on
stuff
there
on there
for
for
bioinformatics.
bioinformatics.
Bioinformatics is
Bioinformatics
such
is
a
such a
wide
wide
field as
field as
well.
well.
exactly.
I'm just
I'm
talking
just
about
talking about
genetics really,
genetics,
but
really, but
bioinformatics
bioinformatics expands
expands to
to
much more
much
than
more
just
than just
genetics.
genetics, and
I would say
would
pick
say
something
pick
that
something that
is
is
related to what
related
you're
to what you're
working on
working on,
in terms
in
of the
terms of the
aspect of
aspect of
biology that
biology
you're
that
working
you're
on.
working on.
I think one has to know biology first and then bioinformatics. That's like my understanding. Though I'm a protein guy.
I
I
think
think
data
data
science
science
skills would be
skills
good
would be
to
good
learn,
to
and that's
learn, and
sort of
that's
more
sort of more
generic.
generic.
But
But
yes,
yes, I
you
think
won't be
you
able
won't be able
to
to
do
do
top-notch
top-notch
bioinformatics if you
bioinformatics if
don't
you
know the
don't know the
biology
biology
well.
well.
I think that's
I
what
think
I'm
that's
trying
what
to
I'm
get
trying to get
across as
across as
well.
well.
It's not
It's
just
not just
about
about
being able to
being able to
program in
program in
Python.
Python.
It's
It's
understanding the
understanding the
problem
problem
from a
from a
biological
biological
perspective.
perspective.
And these questions, what are de facto and C dot and G dot?
So
So
C
C
dot is
dot
the
is the
level of
level of
description.
description.
So it's
So
the
it's the
cDNA
cDNA
level.
level.
So
So
ATG would be the start code and it's with reference to that. So C.2 would be the second base. It would be the T, and this is the location of computation. The G dot is the genomic level. So when you've got your VCF file, you can also use that as the G dot level. So the position, the coordinate is the G dot in the genome.
And what methods do you use to find the probability of disease-causing mutations, coding routines?
So I just based that study where they looked in the coding region, and for 60% they could find disease-causing mutation, but 40% of patients they couldn't find any coding region variant that caused disease. And then I looked at the database we have, seeing the published literature, how many non-coding mutations are reported, that's around 10%. But there is a bias, that's what we've got to be aware of. There's a bias towards publishing coding mutations. So maybe in the future, that bias will sort of even out a little bit as tools develop to interpret non-coding regions. It's difficult.
So I believe that most of the questions answered. And Matt has given me email ID. You can contact for any other queries and doubts. And he's an excellent teacher. He taught me programming to remember. He's
Thank you.
And Matt, would you mind stopping this slideshow? I have something for you.
Oh, thank you. I've stopped it now. Oh, thank you. That's lovely. Thank you.
That's a small thing from all of us. So I'll send that to you by email.
And I'll send you the slides as well. So if anyone wants to copy the slides, you can distribute them.
Okay, sure, Matt. Okay. So thanks a lot, Matt. And we'll be working on the model project, and probably we'll get the results. Because we're moving to the States, there are a lot of things going on. So I'm sorry for that. Probably.
No, that's no problem. No, that's what collaboration is all about, and it's communicating, and there's always times when we're really busy and things can't be done as fast as we would like. There's nothing wrong with that. No, thank you, Matt.
Yes, Matt, and have a good day. Okay. Bye.
Thank you, everyone.
So, information email ID can note করে দিচ্ছি. Please pretty friendly, and you can ask any question if you wish. He's available. আর তোমাদের কয়েকটা question ছিল. I think those questions are not for him. Skills required and suggest some small beginners project. Skill বলতে এখন ব্যাপারটা হচ্ছে it depends on what you want to do. If you want to do মানে ও যেটা দেখালো that is mainly genomics. তো তোমরা যদি genomics করতে চাও এই databases গুলো তোমাদের কনসিডার করতে হবে। আর বেসিক্যালি প্রোগ্রামিং এর দিকটা তোমাদের খুব স্ট্রং। আর যদি প্রোটিওমিক্স এর দিকটা চাও আজকে সকালে যেগুলো আমরা ডিসক্রাইব করছিলাম ওই দিকটাই তোমাদের ওই দিকে কনসেন্ট্রেট করতে। কিন্তু যেটা ও বললো যেটা আমারও পয়েন্ট সেটা হচ্ছে যে বায়োইনফরমেটিক্স এটা হচ্ছে এটা খুব বা ইন্টারডিসিপ্লিনারি সাবজেক্ট যেহেতু মানে তোমাদের প্রবলেমটা ডিফাইন হতে হবে হ্যাঁ মানে তুমি জাস্ট বললে যে আমি একটা বায়োইনফরমেটিক্স করবো সেটা না একটা ডিফাইন প্রবলেম হতে হবে একটা ধরো তুমি একটা কোনো সিকোয়েন্স চলে আসবে সেই সিকোয়েন্স রিলেটেড আমরা কি কি ইনফরমেশন দিতে পারি সেগুলো আমরা তোমাদের দেখিয়ে দিতে পারবো কিন্তু জাস্ট মানে ফর দ্য সেক অফ ডুইং বায়োইনফরমেটিক্স সেইটা করার সেটা মিনিংলেস সেইভাবে যেটা যেটা আমারও ভিউ যেটা উনি যেটা বলবেন সেটা হচ্ছে যে এটা এতটাই ইন্টারডিসিপ্লিনারি যে বেসিক নলেজ যদি ঠিক না থাকে তাহলে খুব একটা প্রসিড করাটা একটু ডিফিকাল্ট তো স্কিল সেট বলতে তোমাদের আমি যেটা বলবো বা সবাই যেটা সাজেস্ট করবে সেটা হচ্ছে যে প্রোগ্রামিং এর দিকটা যদি একটু তোমাদের জানা থাকে আর স্ট্যাটিস্টিক্স আর প্রোগ্রামিং এই দুটো বেসিক্যালি এখন মানে বেসিক সাবজেক্টের মধ্যে এইগুলো আর তাছাড়া সবার ব্যাকগ্রাউন্ড যা যা যেরকম আছে কেউ একটু বায়োলজি পড়ো কেউ বায়োটেকনোলজি পড়ো কেমিস্ট্রি পড়ো সেই বেসিক ব্যাকগ্রাউন্ডে তো থাকবেই আর বিগিনার্স প্রজেক্টস যেটা বলছো বিগিনার্স প্রজেক্টস বলতে যেটা যেভাবে তোমরা স্টার্ট করতে পারো সেটা হচ্ছে যে আজকে ধরো তোমাদের কোনো একটা পিডিবি দেখানো হয়েছে সেইখানে পিডিবিতে গিয়ে তোমরা কোনো একটা পার্টিকুলার অর্গানিজমের নাম দিয়ে কোনো একটা প্রোটিনের নাম দিয়ে সার্চ করে দেখতে পারো এবার সেখান থেকে তুমি কি কি ইনফরমেশন পাচ্ছ কি কি প্রোটিন আইডি পাচ্ছ সেই প্রত্যেকটা প্রোটিনের স্ট্রাকচারস গুলোকে দেখতে পারো প্রত্যেকটা প্রোটিনের লাইগেন্ড যেটা তুমি বলছিলে যে লাইগেন্ড আছে কিনা এবার সেটা ওর এক্সপ্লিসিটলি ওরি লাইগেন্ড নাকি ওটা হিস্টোরিওগ্রাফিক লাইগেন্ড সেইগুলোকে আইডেন্টিফাই করতে পারো সিকোয়েন্সও দেখতে পারো সিকোয়েন্সটা একদম একদম মানে ওয়ান থেকে স্টার্ট হয়েছে কিনা সেটা দেখা যেতে পারে তারপরে ওইখানের সাথে লিঙ্ক করে তোমরা ইউনিপ্লটে গিয়ে সিকোয়েন্সের সাথে মানে ম্যাচ করে দেখতে পারো যে কোনো মিউটেশন আছে কিনা প্রোটিনটার মধ্যে কোনো ওর পোস্ট ট্রান্সলেশন মডিফিকেশন হয়েছে কিনা বলে কোন পার্টিকুলার অ্যামিনো অ্যাসিডের মধ্যে হয়েছে এই এইভাবে স্টার্ট করতে পারে বিগিনার্সদের জন্য বলছি আর যদি রিসার্চ যারা করবে তাদের তো ডেফিনেটলি যেখানে যে ল্যাবে যাবে সেই ল্যাবে স্পেসিফিক প্রবলেম তো থাকবে হ্যাঁ সেই ল্যাবে তো হোপফুলি তোমাদের কিছুটা আজকে ক্লিয়ার হয়েছে ব্যাপারটা মানে আমরা আবারও নেক্সট থেকে আমাদের যখন নিরাজনা সেনগুপ্ত আছেন উনি মেইনলি ডিসকাস করবেন প্রোটিউলিক্সের উপর আমরা এখানে তোমাদের চেষ্টা করছি যে আজকে আমরা একটু সিকোয়েন্স লেভেলে আমরা একটু দেখলাম একটু স্ট্রাকচার একটু সিকোয়েন্স লেভেলে দেখলাম কালকে একটু স্ট্রাকচারাল লেভেলে আর একটু প্রোটিউলিক্স লেভেলে আমরা দেখবো আর কালকে আমরা আবার একটা কনসার্ন আমাদের আছে আর কালকে সেকেন্ড হাফে যে টপটা আছে তিনি বলবেন উনি খুব ইন্টারেস্টিং একটা টপিকের উপর উনি ডিসকাস করবেন যেটা হচ্ছে যে ট্রান্সক্রিপশন ফ্যাক্টর ট্রান্সক্রিপশন ফ্যাক্টর সবারই জানা আছে ট্রান্সক্রিপশন ফ্যাক্টর কি সেই ট্রান্সক্রিপশন ফ্যাক্টরের ফাংশনালিটির উপর উনি ডিসকাস করবেন ওনার একটা টুলও আছে সেই টুলটার উপর উনি কথা বলবেন ট্রান্সক্রিপশন তো বুঝতে পারছো যে একটু ইন্টেন্সিভ বায়োলজি হয়তো সেখানে ডিসকাস করা হবে আর as well as other biology ঠিক আছে সো তাহলে সবাই কালকে 11টা থেকে স্টার্ট হচ্ছে হ্যাঁ সবাই চলে এসো একটু মানে ধরো উইদিন 10 থেকে 45 আসলে ভালো হয় কারণ ওখানে আর কি সেটা করে আর কি তোমাদের ইনক্লুডেড করতে যেতে একটু টাইম লাগে এখানে একটু ব্যাড করবো উইদিন 10:45 যদি সবাই চলে আসো ভালো হয় তাহলে আমরা এখানে স্টার্ট করব আর এই বিল্ডিং এখানেই আসতে হ্যাঁ এই বিল্ডিং এখানেই হ্যাঁ প্রোগ্রামিংটা সেটা অনেক বড় ব্যাপার কোনো টুল ইউজ করা I'm interested in the sector তার সঙ্গে যে বিজনেস আছে তাদের রেগুলেটরি ফাইল সেখানে কিভাবে বাইন্ড করবো ট্রান্সক্রিপশনটা এই টাইপের জিনিসগুলো দেখার জন্য আচ্ছা একদম তোমাকে কোডিং থেকে সার্চ করে সেখান থেকে দেখতে পেলাম সে ইন্টারনেটে কিছু ইনফরমেশন আছে এবং সেখান থেকে তাকে যে ইনফরমেশন পিডিবি আইডেন্টিফাই তার স্ট্রাকচার তার আইডিয়াকে ওখানে ওইখানে থেকে দেখতে পাবে যে মেটাল আছে তুমি যে মেটাল আইডেন্টি ধরো তুমি ম্যাগনেসিয়াম বা ক্যালসিয়াম তোমাকে ক্যাডমিয়াম ডেফিনেটলি ওখানে দেওয়া
নেচারালি যেভাবে মানে দেখানো যাবে সেটি স্ট্রাকচারটা পাবলিকেশন আমি প্রোটিন তার সঙ্গে লাইটের ইন্টারাকশন আচ্ছা এখানে হচ্ছে বায়োমেট্রিক সেন্টার কিভাবে ইউজ করা হয় সিস্টেমটা অনলাইন হ্যাঁ হ্যাঁ প্রবলেমটা এইগুলো এগুলো করা আজকে শুধু ওয়ার্কশপ আমি আজকে নেক্সট উইক আমি একটা সময় করে তোমাকে জানিয়ে আসবো
তো কালকে তাহলে সবাই ভিজিটিং করতে পারে এখানে চলে আসে এখানেই আসবে তো সার্ভ আউটসাইড
সরি আপনি বোধহয় একটু শর্ট মিস করে গেছেন তখন মনে হয় আমার ডিলের জন্য মনে হয় মেল আইডিটা দিলে আমি পাঠিয়ে দিতে পারি আপনাকে আমার আসলে ক্যামেরাটা বন্ধ করছিল ঠিকঠাক আসছিল না
যে জায়গায় কাজটা সেটাতে মিউটেশন জেনেটিক ভেরিয়েন্স পিএইচডি কমপ্লিট হয়ে গেছে অনেকদিন আগে এখন আবার একটু মাঝখানে কিছুই করছিলাম না তারপর
দিদি আমার সাথে ঘুরতে যাওয়ার প্ল্যানিং করছে তো যাবি কি করে সেলফি হয়ে গেল আমি পারি না
আরে দাঁড়া আমি 5 মিনিট বাদে ফোন করি
যখন এসে দেখো ফোনে ব্যস্ত
জীবনে এইটা ছাড়া এখান থেকে একবার চাই দেখে আসি না না আমি দেখে নিচ্ছি
ওটা সবাই কিন্তু চাপতে সবাই চাপ চাপ সবাই দিয়েছে তো পরে নিতে পারবে আচ্ছা
তো সিস্টেম খোলা আছে
এইটা আমি তো ওখানে
না
নিয়ে
আমার
গেছি
কাছে আছে
ও তো এখানে আছে দিদি একটু যাবে ডিপার্টমেন্টে তাহলে